So we've talked about a particular type of non-covalent interaction, that is the hydrogen bond, and its relation to water. But let's focus a little bit more on other non-covalent interactions and their relation to biochemistry and water. Again, one of those is the hydrogen bond. This is where you're going to have a hydrogen with a partial positive charge interacting with some electronegative or electron pairs off of an electronegative atom, typically oxygen or nitrogen. There are also ionic interactions. This is where you have two molecules with full charges, a positive charge and a negative charge that are pulled together because of these oppositely attracting charges. You can also have ionic interactions that are repulsive as well. We typically only think about interactions when they are attractive and forming bonds, but interactions also include repulsion interactions. So two positively charged ionic molecules will repulse each other and actually form more distance than usual. Another type of non-covalent interaction we will focus on a little bit later is the hydrophobic interaction. This is going to occur when you have hydrophobic or water-fearing things that do not get along with water very well. These types of molecules crowding together when also in the presence of water. A very good example of this happening is when you drop oil into water, and that oil is going to crowd together into one big droplet instead of just dispersing through the water. And lastly, another non-covalent interaction that is often overlooked but still very important is the van der Waals interaction, which just is a proximity interaction where you have the surface area of two molecules actually causing some interaction, either a attraction or repulsion, just based on surface area proximity. An example of these non-covalent bonding interactions is the dissolving of a salt. So dissolving involves breaking of one type of non-covalent interaction, which is an ionic interaction. Salts are held together by positive and negative atoms that are interacting. So that's an ionic interaction. And these are broken down and solvated by water. Water surrounds each of those individual ions, forming instead this dipole charged interaction in which the dipole of a water molecule, the partial charge of a water molecule, is interacting with the full charge of an ion. And you can see in this picture, and it's noted that the water molecules are not just randomly orienting themselves around each of those ions. Notice that for the chlorine, which is negatively charged, the hydrogens are pointing towards it. Hydrogens are partially positive. So we have a partial positive and a negative interaction. Oppositely, on our sodium, which is positively charged, the oxygens are pointed towards it. Oxygens are partially negatively charged. Again, you have a partial negative and a positive interaction. Now let's talk about van der Waals interactions a little bit. Van der Waals interactions have two components. As I noted, there is an attractive component to it and also a repulsive component. For van der Waals, these are names. They're given a name. The attractive force is called London dispersion forces. And the repulsive force we often refer to as steric repulsion. Attractive forces are dependent on polarizability, the ability of a molecule to become polarized, meaning that it doesn't necessarily have a dipole, but it can be forced to have its charges redistributed somewhat to form some polar interactions. Just a slight redistribution causing it to become polarizable. Repulsive forces, steric repulsion, often depend on the size of the atom, and that is going to determine its steric interactions. So let's look at an example of a van der Waals interaction. It's actually one potentially everyday item that some of you have, which is a microfiber cloth. Microfiber cloths actually work through van der Waals interactions, and they do it through surface area. You can see on the left, there is a picture of a microfiber cloth fiber, essentially, which is just a combination of different polymers, but they are made into this super fine strands with very, very small diameters, about 50 times thinner than a human hair. And these very small fibers, which are all throughout the cloth, you can see as such, have more potential to have these van der Waals interactions through their surface area with dirt molecules. This is opposite of something like a very large, bulky fiber, which cannot have these interactions except maybe on the surface. But having more surface area by having those thinner fibers causes more van der Waals interaction. These are very weak individually. Each one van der Waals interaction is not strong. So it is easily broken and reversible. But when you have many van der Waals interactions, you can get some strength out of that. For example, how geckos can climb walls. They're actually having thousands of interactions of these van der Waals interactions on their little toes that can then cause some strength in numbers. This interaction is pretty universal because it can occur between any two atoms that are just near each other. And it is related to a lot of biological interactions, which is why we're bringing it up here. Again, it can actually stabilize molecules. So an example of this in a biological molecule is in DNA. When DNA forms, it actually has some stacking that occurs as it builds. And this stacking causes surface area of molecules near each other, which then is related to van der Waals interactions. Now, one very important non-covalent bond that was mentioned is the hydrophobic effect. This isn't necessarily a bond per se, but it is an effect related to our non-covalent bonding. It actually refers to the association or interaction of nonpolar molecules or components of molecules that happen to be nonpolar when they're in an aqueous solution, so generally in water. It is a huge driving force in biochemistry behind things like protein folding, protein-protein association, how they actually interact, the formation of lipid micelles, which is related to how cells form their membranes, and even things like binding of steroid hormones to their receptors within the body. Now, again, I want to be clear, this does not arise because of some direct force. There is not a force that actually is a nonpolar force holding these together. This is an effect that's occurring situationally, in fact, because of entropy. So we'll look at this. When water surrounds something that is not polar, that does not get along with water, it does not dissolve well in water, it causes a change in entropy. So you can see here, water is generally highly ordered in this situation. So it actually causes a lower entropy to occur when you place in something with nonpolar regions. So this is an example of a lipid tail, and the yellow is something that is nonpolar, hydrocarbon, does not dissolve in water, cannot interact with water through its dipole interactions or hydrogen bond interactions. And so it causes water to cluster around, but it can't actually interact. This causes low entropy. Again, this is actually more ordered than usual. Water has to cluster in this way around this individual molecule, and that is very unfavorable. Thermodynamically, if we think about it, entropy likes to be high. It likes to be disordered. Your room likes to get messy. It doesn't like to get cleaned. So if you had a lot of lipids in water, so here is an example. We have many different little lipid tails sitting in water. We now have many areas where water has to form this ordered structure, causing low entropy in those areas. This is going to, over the whole system, decrease entropy, which is again a very unfavorable state. Now, all of these molecules of water that sit around each individual lipid is in this ordered and unfavorable state. Now, if these nonpolar portions were to aggregate, cluster together, fewer water molecules are now actually ordered. Now, there are still some, those ones that surround the outside have to be ordered. They can't interact exactly, and they're kind of stuck in this ordered position. But overall, there are less total water molecules than when we were looking at them here in this state, separate individual pockets. Overall, this causes entropy to increase. It allows water to be a little bit more disordered, which is a more favorable situation. This entropic driving force, entropy itself, is what drives the hydrophobic effect. It's what drives these nonpolar groups to cluster together so that it is a more favorable entropic state for water. This is also what helps to form things like micelles. When you have a molecule that has a hydrophobic end and a hydrophilic end, this entropic drive will continuously drive that hydrophobic portion to cluster. Now, the hydrophilic end, that head group, happens to be polar and allows for interactions with water. This side then tends to face the water. It is having interactions with the water, and we again have driven that hydrophobic portion to cluster together as far away from the water. This state, when you happen to have something that has a polar end and a nonpolar end, is the most favorable situation and causes micelles. This is when, again, all of the hydrophobic groups are sequestered inside, and the whole outside is just the outside facing polar groups, which interact nicely with water. So just let's look at one example of how the hydrophobic effect is important in biological molecules and in biological systems. This is the hydrophobic effect and its relation to ligand binding. This happens when we have some substrate, some molecule that needs to bind with an enzyme. This is occurring all the time in our bodies. The binding site of an enzyme is often hydrophobic, and the substrate portion that needs to bind, again, is oftentimes hydrophobic. So these entropic interactions, natural hydrophobic effect, will force the side of the enzyme and the side of the substrate that are hydrophobic to cluster together. Again, it's just entropy that are driving these areas of hydrophobic in both the substrate and the enzyme to then cluster together. This is a design advantage when making drugs. If we know that there is an enzyme that happens to have a hydrophobic binding pocket, then it is desirable to make a drug that also has a hydrophobic portion so that it will then interact. Entropy will drive them to interact naturally and form this needed binding between the enzyme and the substrate.